Hello, everyone. Welcome. It's good to see everybody today. Welcome, welcome. We've got a couple minutes before class time, so we'll give everybody a couple minutes to join in. If you would, go ahead and turn your video camera on, and you can leave your microphone muted. That generally helps us stream more smoothly. So, yeah. And if you would, go ahead and type your nickname or your first name into chat for me. That'll be how I'll take attendance today. Just go right ahead and type your first name or a nickname if you have one right into the chat for me. Thank you. Thank you very much. All right, very good. It's good to see everybody. I hope your day is going well. So we are remote this week, but hopefully that's a good thing. Hopefully that will allow us to keep everybody as safe as possible, and it'll allow us to get back together next week in person. Okay, a couple more people are coming in. Yeah, just type your nickname or your first name into chat for me. That's how I'll take attendance today. Very good. Okay, well, we're just at 3 o'clock, so I'm gonna go ahead and get things started. My name's Dr. Piscapo, and I'm gonna be your instructor for cell biology this semester. If you've not had me before, I'll tell you just a little bit about my background so you know where I'm coming from. I'm a veterinarian. I grew up in Massachusetts. I know some of you are from Massachusetts. I went to college in Vermont. I went to the University of Vermont and I studied animal science. And then I went to veterinary school. I went to North Carolina State to go to veterinary school. When I graduated from there, I went into private practice. I actually stayed in North Carolina for about 10 years. I had an opportunity then to go back to school. I was offered what's called a fellowship to go back to school and earn a PhD in human physiology. So, so I have that as well. I actually suffered an injury to my spinal cord at one point, which kind of forced me to retire from practice. That's how I got into teaching. And I moved back up to New England and started working for Keene State. Gosh, way back in 2008. So I teach quite a number of courses here at Keene State. I also teach at other schools, other colleges in New Hampshire. I'm what's called an adjunct professor, and basically that means I'm here part-time. I teach at Keene State part-time. I teach biology and evolution, and I teach cell bio and genetics and microbiology and all kinds of different things. So I'm happy to be teaching cell biology this semester. It's a great course. And I'm glad to see everybody here. I'm not going to go through our syllabus and schedule with you today. I think you can do that on your own. Everybody in here is a second-year student or above. So I think you can read through a syllabus without too much difficulty. If you go on our Canvas course, you know there's a Start Here module for you to work your way through to sort of get introduced to the course. Many of you have already done that. Thank you. There is a survey for you to complete for me in that Start Here module, a student survey. I give those surveys out at the start of every semester in order to help me get to know you a little better. It helps me to sort of gear the class towards your interests and your plans and things. So if you haven't already completed that survey, go ahead and get that done for me by the end of the week. And that's right in the module on Canvas called Start Here. What we will do today is we'll have our very first lecture about the universal characteristics of cells and a little bit of cellular biochemistry. And I do want to talk just a little bit about some organizational things that are happening in the course this semester that are a little different for us. We are a big group this semester. We are a larger group than I ordinarily have in this class. So in order to accommodate the laboratory exercises, we're actually going to be splitting into 2 groups for laboratory. So we'll all have lecture together on the days we're scheduled for lecture. And then we will split on days that we're doing laboratories. And what that will mean is about half of you will be doing the laboratory on that day, and the other half of you will be watching a lecture video that I've posted for us on a playlist on YouTube. So I have a channel on YouTube that has lecture videos posted to it, and I have a playlist that is dedicated to this class. So when we look at our schedule, you'll see that the labs get run twice, 2 days in a row. Half the class takes it on the first day, half the class takes it on the second day. And the other half of the class views a lecture from home, a recorded video lecture from home. That will allow us to stay in small groups in the laboratory, which is much more workable for us. So this class is, it's designed to follow the in BIO 110 class. Now, some of you are coming straight out of in BIO 110, and some of you are about a semester away from that. You took evolution last semester. That's fine as well. It's not going to cause any problems for you. But we do a fair amount of review in this course because there are certain concepts in biology that it's always helpful for us to review. So especially at the start of the course, you're going to notice that some of the topics that we're discussing are similar to what you talked about in BIO 110, cells and molecules. But you're going to notice that we go into a little more depth now. We go into a little bit more detail about some of the basic processes in biology. As we move forward in the semester, we'll start into some topics that are brand new, things that we haven't talked about yet in the biology curriculum. And that will come again more towards the middle of the semester and beyond. So let me ask first if anybody has any questions about what you looked at on Canvas, if you've gone through that Start Here module. If anybody has any questions about the syllabus or the schedule, if anybody has any questions about course policies, feel free to type your question into chat. I think it's pretty straightforward, but we can certainly deal with questions as we go forward in the semester as well. We can talk about, for example, we can talk about examinations and what they're going to look like as we get a little bit closer to them. So feel free during our meeting today to type any questions you have right into chat, and we'll address them. So the course is organized completely on Canvas. Everything that I use in the course will be available to you on Canvas, including all of the PowerPoint lecture slides that I use. It is important that you take good notes in this class, but understand that you have the slides available to you at all times. So when you're taking notes, you don't need to write down every single thing that you see on the slides, because that would just be extra work for you. Many of my students, in fact, bring a laptop to class and they open up the slide set that's available on Canvas, and they use the notes section underneath each slide in order to take notes during class. Other students who prefer to handwrite their notes, they may do something like in their notebook, they'll write slide number 1, and they'll just jot down any additional things that I'm saying about a slide. Just don't try to write down every single thing on every single slide during our lectures, or you'll drive yourself crazy. You have the slides, you have all the information that's on the slides available to you on Canvas at any moment. So you don't need to worry about writing everything down, just the extra things that I say during lecture. So Sophia's asking about lecture outlines, because one of the assignments that I'm going to give you this semester regarding lecture is I'm going to ask you to outline lectures so that I can see that you are taking a lecture and doing something with it at home, so that it's in a much more compact form, so that you can study the material much more efficiently. My lecture slides are actually quite complete. And by that, I mean, most of the information that you're going to need to know for an exam is right on the lecture slide. I do say additional things about the slides when I lecture, and you'll want to jot those things down. But most of the information is right there on the slide. It's just that you're going to need to take those lectures and create some kind of study materials for yourself that you can then memorize for the exams. It's not a good idea to prepare for exams in this class by just looking through the slides. You're going to want to make some kind of study material for yourself so that you can memorize that document or that stack of flashcards or whatever other kind of study materials you like. So Sophia, when I assign a lecture outline, when I ask you to outline a lecture as an assignment, you're going to do that however you see fit. However you see fit to outline that lecture material, so that you take those, let's say 30 slides, and make yourself, let's say, a 3-page outline. That's going to be a lot easier for you to use to study. It's a lot easier for you to memorize a 3-page outline than for you to memorize 30 slides. Now, if you're familiar with PowerPoint, you know that you can take a PowerPoint lecture, you can open up a PowerPoint lecture, and in the menu up top, you can view that lecture as an outline. You can click on Outline View. And what will happen is PowerPoint will take those 30 slides and put them into an outline for you. You're going to have to do a little cleanup on that outline, because it will include things like the titles at the top of slides and things like that. It'll include some information that you don't really need in the outline. But yeah, if you'd like to try that, if you'd like to create the outline directly from the PowerPoint slides and then sort of customize it for yourself, you're free to do that. I just want to see that you're making some study materials for yourself. And some of the most effective study materials that I find for this material are just plain old, good old-fashioned outlines, where you put the information into an outlined form with indentation, main headings and subheadings and so on and so on. And then again, that will be available to you when it comes time to study for the exam. And you'll learn a few points. You'll learn a few points for doing it. Good question. So Sam is asking if we can just submit our notes instead. Sam, when I assign an outline, what I'm really doing is asking for what you're going to do with your notes. You may have experienced in previous classes, you may have found that by going through the lecture and reading your notes, that was a good way to learn the material. But I think you're going to find that in this course, that's not a very efficient way to prepare for an exam. So one of the things we try to do is encourage you, to try your hand at making study materials. And again, all I mean by study materials is some kind of a document or some kind of a, again, some students use flashcards, something where you've taken this big volume of material and you've turned it into a much smaller volume of material, a much more condensed, much more efficient set of facts that you're then going to study and learn and memorize for the exam. So to answer your question, Sam, no, I would not want to just see your notes. I want to see what you're going to do with your notes and your the slides.
Okay, because my notes that I usually take are like very organized and outlined, kind of like a study guide.
Oh, sure. Well,
Okay.
if you outline your notes, then you
yeah, like if they're
absolutely
usually like
submit
extremely
those.
organized.
Yeah. A lot of students, what they'll do is when they go home, they rewrite their notes, and they often outline them. And if you're one of those students, that would be what you would want to send me, what you would want to submit to me when I assign that kind of thing. It's really important to learn this technique, to learn how to take a large volume of information and condense it down into just the bare bones, the key facts, the things that the lists of things, and the vocabulary words and the things you need to memorize. It's a skill that you can use in any class, a science class, a humanities class, any class at all that you are tasked with learning information. If you practice at making study materials for yourself, and you get good at it, you will find that it will help you a lot in terms of getting that information committed to your memory. The other thing is research studies have shown that the act of making study materials, the time that you put in to making an outline or making flashcards, that alone helps you learn. The fact that you are taking the information from one form and putting it into another form is helping your brain commit it to memory. So that's why we're doing that in this course, to encourage you to use this kind of a technique, because it's something you'll be able to carry with you as you go forward in your educational career. It's a really good technique to learn. I see Cole is asking about the instructor interaction plan. It won't let you view or download it. It says you do not have access. Oh my, that sounds very important and official. I will double check that, Cole, after class. Must be a bad link. Thanks for letting me know. So Sam's asking about submitting flashcards. If you use flashcards, listen, if you find flashcards helpful, I want you to absolutely make and use flashcards. But when I assign you to make an outline, I want you to try the outline. I want you to try doing that. If you make flashcards for yourself as well, that's fine. But I want everybody to try this sort of an exercise, an academic exercise of outlining lectures periodically. It won't be for every lecture, but periodically I'll ask you to do that. And again, if you find it a daunting task, remember, you don't have to start from scratch. You can open up the lecture, you can open up the slides, and then you can click on that menu option, that toolbar option, to show the slides in an outline view. And then what it'll do is it'll take those 30 slides and it will put them in an outline. You then have to go in and, you know, clean it up a little bit. But it's a starting place for you if you're not comfortable just jumping in and starting your own outline. Good question. Yeah, so Julia, same thing. I won't want a Quizlet link or anything like that. These will specifically be outlines that I ask for during the semester. And again, it won't be for every lecture. It'll just be periodically. Much of the time, instead, what you'll be assigned is just a homework. I'll ask you to complete a homework about the lecture to make sure, number one, that you're understanding the material, and to give you an opportunity to see the kinds of questions I ask. On those lecture homework assignments, you'll get a chance to see the kinds of questions that I typically ask and how I ask them, what kind of language I use. You're going to find that most of the questions that you'll get from me in this course are going to be multiple choice questions, but I use a whole variety of different formats for multiple choice. So for example, I use true false questions. I'll ask you whether or not a statement is true or false, and why it's false. And you'll have to pick the correct reason from a list. I ask you to fill in a missing term from a statement. Choose the term that best completes a statement. Sometimes I ask you to pick one term out of a pair of terms. Which term would be correct in this situation? I ask a whole variety of different kinds of multiple choice questions. So you'll get a chance to see how I write questions when you complete the lecture homeworks. And then you'll be better prepared, hopefully, for when the exam comes. Samantha's asking, do we need to, can we handwrite an outline or does it need to be typed? I actually do accept handwritten outlines. The only difference is that you would need to take a picture of the outline and submit it to me. I wouldn't want your actual outline. I would just want an image of your outline. But yeah, I'm happy to take handwritten outlines because again, studies have shown, research has shown that when you handwrite, you're presenting the information to your brain in a way that helps your brain learn it. So I encourage students, if you like to handwrite your outlines, you should. It may actually help you memorize the information. Yeah.
I have a question.
Yes.
So if we're taking pictures of our outlines, like how would we turn it in? Because like in the
Oh,
past for like pictures and like other classes, it would be like a Google like doc you would put the photos onto. Like, is
yeah.
that fine?
God knows there are all different kinds of ways to submit things like that. Generally, what I have students do is take that image and insert it into a Word document. So open up a Word document and do a copy and paste of the picture, and then submit the Word document through Canvas. Remember, as a Keene State student, you have full access to Office 365 on the Keene State website. And the Office software includes Word and Excel and PowerPoint and Outlook. So you always have Word available, Microsoft Word. And again, that's the way I typically do it. I have students put their picture right into a Word document and then submit that into Canvas. Good question.
I just wanted to mention there is an app called OneDrive where you can like take a picture of your document and it scans it and makes it clearer and it connects right to your email, and then you just open your OneDrive email and it saves in there and you can upload those
Oh yeah, like
to Canvas.
there are lots of ways, as you all know, as we all know, there is lots of technology available to us now, lots of software, lots of applications. It's crazy. And what you're going to find is that from class to class to class, different professors have different preferences about what they want you to use. But it's all good experience. It's very good experience to try different things and try different software and get some experience doing that. It'll help you in your professional life if you have that kind of experience. So, all right. So that looks good. All right, very good. So I'm just going to pull up Canvas for just a minute so we can take a look at our course on Canvas. That's what you should be seeing in just a sec when I clean this up. Let me do this one more time just to be sure. All right. So on your screen right now, you should see our home page on Canvas. It says cell biology across. This is the home page. This is what I like to think of as our home base. If you click on home in the menu, you'll come to this page that has all kinds of information that you may want to refer to again and again and again as you go through this semester. For example, on this homepage, there's always going to be a link to the syllabus and schedule. And if I need to update anything in our schedule, I'll do that and post the syllabus here. So you'll get a nice, fresh, up-to-date copy. We have Zoom information on this page. And you also have a link to the YouTube channel and the specific playlist for this course. So I create video recordings of lectures and I post them onto this YouTube channel called Science Science in a dedicated playlist, a playlist that is just for our class. Typically, a lecture is broken into between 1 and 4 videos. So there'll be, 1 of 4, and then 2 of 4, 3 of 4, 4 of 4. That way, none of the videos are very long. There's a lot of lecture videos already posted in that playlist. More will be added as we go this semester, but that's going to be a place for you to find recordings of lecture videos. Again, when our schedule asks you to watch a lecture from home, when we split up to do laboratory activities and so on. I do want to just remind everybody that we are on campus this semester. We are in person this semester. And what that means is that you are expected to come to class and do lectures in person in the classroom with me and with your classmates. Obviously, there are going to be times when some students cannot come to class because of illness or because of personal family emergencies and things like that. If you find that happening to you, if there is a day when you cannot come to a scheduled class, you're going to want to notify me. You're going to want to message me specifically through Canvas. You're going to want to send me a message through the inbox function in Canvas and let me know. Let me know that you can't come to class. That way I'll know you're not going to be there. And I'll know that you'll be viewing a lecture from home or something along those lines. I can also, if you're excused from class, I can offer you an extension on assignments if those are needed. So just be sure that if you need to miss class, that you message me. Now, if you go over into this menu on the left-hand side, and scroll down it, you can see all these different links for functionality in Canvas. There's a second menu. There's a far left menu next to it. That's where you get to the dashboard and so on. And this link right here is called Inbox. So if you click on that, you can send me a message through Canvas. Now, there is a reason that I ask you to use the Canvas inbox. And that is that it sends student messages separately from all the rest of my email. So it helps me to see your messages right away. I can get, upwards of hundreds of emails on any one day. And it's easy for me to miss a message from you if you send it through our regular Keene State email system. In comparison, if you message me through Canvas, I get those emails completely separate. So I can really focus in and see your messages much more quickly than if you send them to me through traditional email. So anytime you want to send me a message, do it through Canvas, and it will come right into that special mailbox for me. Now, we have had a change in our COVID policy this semester. I'll just briefly mention, we're taking into account this semester whether or not you're vaccinated in terms of getting a positive test, right? The policy, of course, is available to you on the Keene State webpage. I'm sure you'll hear it more times than you care to this semester. But the important thing to note this semester is that 2 things. Number one, if you are sick, stay home. All right, that message hasn't changed. If you're sick, stay home. Send your instructors, all your instructors, a message. Tell them, I'm sick, I'm staying home. All right, and again, in this course at least, you'll have access to that YouTube playlist where you can watch the lecture that we're doing that day. Now, if you miss a lab, it'll be a little different because there aren't video recordings of our labs. But, you know, you can get in touch with a classmate and get notes from the classmate, and you'll still be able to do the assignments associated with the lab. Where the new change comes in is when you test positive during our surveillance testing, our weekly surveillance testing. Remember, if you are vaccinated and you are asymptomatic, you have no signs, no sickness, a vaccinated asymptomatic person who gets a positive test in surveillance testing can still come to class. You just want to make sure you're all masked. I mean, we're all going to be masked anyway. So that's a nice change because, of course, we have enough data now to show that COVID is a very different disease in vaccinated people versus unvaccinated people. So if you're vaccinated and you have no symptoms, but you test positive, you're still welcome to come to class this semester. You're just going to want to monitor yourself. And if you start to develop any symptoms, you're going to want to stay out of class. Unvaccinated people who test positive, they're still going to need to isolate themselves at home. Quarantine themselves at home, whether they have symptoms or not. Okay. I know that, Caitlin, I can see your message. I did receive your email. Thank you very much. The really important thing to remember this semester, not just for this class, but for all your classes, is you really need to stay in touch with your instructors. It's a hectic time for all of us because we're trying to have a semester. We're trying to have a learning period, and we're also dealing with the pandemic. So communication is really, really important. It's important that you don't miss class and not tell anybody, because we don't know where you are. We don't know if you're ill. We don't know what's going on with you. So stay in touch with us if you need to miss class. All right. Any questions? Any questions about anything that I've been talking about? Each week during this semester, there will be a module on Canvas. Each week, there is a module devoted to that week, and all of the materials for that week can be found in that module, including a to-do list. Put a star in your notes that, you know, at the beginning of the week, you're going to want to check your to-do list for this class. Just click on the to-do list and it will summarize for you all of the things that are happening in our class that week, so that you don't accidentally miss an assignment or a due date or anything like that. All right, at the start of the week, find that week's module, click on the to-do list, and set up your own personal schedule for the week around what's happening in our class. All right, very good. All right, well, I'm gonna share my screen again and pull up our first lecture topic, which is universal characteristics of cells and cellular biochemistry. So again, you have these slides on Canvas. One thing that I want you to notice is for my slides, at least my PowerPoint slides, I sometimes put my own notes down here. There's none associated with this title slide, but occasionally you'll see some writing down here under notes. And those are just a few extra things that I want you to have available to you in your notes on your slides. so that you have that information while you're studying. This is a cell biology class, so the very first thing we need to do is talk about what a cell is, remind ourselves what a cell is. And if you remember, a cell is a membrane-bound compartment. It's a membrane-bound subunit of all living things. So if it is a living thing, it is composed of cells, at least one cell, and maybe multiple cells. All of the living things on our planet are cellular. This term cell historically came from some work that was done by a scientist named Robert Hooke. Hooke had a very primitive microscope that he was able to use, and he took a piece of bark off of a cork tree and examined that bark under his microscope. Now, his microscope could magnify up to about 30 times. A modern light microscope can magnify, 1,000 up to 2,000 times, depending on the scope. So he was looking at a much less magnified image than what we could see today. But even then, he could see that that tree bark, that cork bark, had little compartments in it. And to his eye, they looked like little rooms. And back in the mid-17th century, that little room had a name. It was called a cell. That terminology comes from monasteries. In Robert Hooke's day, many gentlemen entered the monastery in order to become religious men. And the monks, as they were called, who lived in the monasteries, were each assigned their own private little room. And it was referred to as a cell. So each person had their own cell where they had their bed and, some sparse furnishings. So Hooke decided to call the compartments inside of the cork bark that he was looking at cells. And to this day, we still use that term to describe these units, these subunits of life. They're referred to as cells. Now, a little bit later on, same century, another scientist who has a terrible name, his name is Leeuwenhoek. That's how you pronounce that last name, Leeuwenhoek. Leeuwenhoek was also looking at cells, but he had an improved microscope. He could see up to a 300 times magnification. And Leeuwenhoek actually looked at a much greater assortment of cells than Robert Hooke did. He looked at things like blood cells, red blood cells, and white blood cells. He looked at sperm cells and algae. He even looked at certain kinds of bacteria under his microscope. So throughout the 17th century, we had multiple scientists, natural scientists, describing the cell and what a cell looked like inside of a living organism. That led to ultimately the production of what we now call the cell theory. And again, we talk about cell theory in Bio 110. The cell theory has two parts to it. All organisms are composed of cells, and all cells come from pre-existing cells. So remember, when I use that word organism, I'm generally talking about a living thing. All living things are composed of cells. If it's alive on our planet, it's cellular. It has at least one cell in it and maybe many cells. And all of those cells had to come from other cells, previously existing cells. In other words, cells don't just assemble out of the air. A cell comes from another cell through the process of cell division. That parent cell, if you will, divides and produces offspring. It produces new cells. And all cells are produced through that process. Now, that process occurs differently in different organisms, but the idea is the same. You can't create a cell out of thin air. It has to come from another cell. And what that means to us is that all of the cells that are on our planet today have come about through evolution across the billions of years of time that life has appeared on this planet. Now, remember, when we consider living things that are made out of one or more cells, we can divide all living things into one of two types of cells: prokaryotes and eukaryotes. Prokaryotes, or prokaryotic cells, are single-celled organisms that have no organelles inside of them and also have no what's called a true cytoskeleton. Eukaryotes, or eukaryotic cells, can be single or multi-celled organisms. Eukaryotic cells do have organelles inside of them, and they have a true cytoskeleton inside of them. So two kinds of living things on this planet: prokaryotes and eukaryotes. Both kinds are cellular because all living things are made of cells. But the cells are quite different between those two categories. Prokaryotes are single-celled organisms. They're made out of one cell. It's a microscopic organism because it only has one cell. And that cell has no organelles in it, no membrane-bound compartments inside of it. It also lacks a cytoskeleton. We don't think about cytoskeleton all the time when we talk about cells, and that's because the cytoskeleton is something we don't see a lot under the microscope. The cytoskeleton, the cellular skeleton, is very important, critical to the survival of a cell, but it's not something we often think about because we don't often visualize it under the microscope. It's hard to stain. It's hard to color that cytoskeleton, so we don't often see it. But only eukaryotes have a true cytoskeleton. Prokaryotic cells, they have something like a cytoskeleton, but it's very primitive. It's not nearly as extensive as the eukaryotic cells have. Remember that when we talk about living things, we can categorize them in other ways as well. We can certainly categorize them based upon traditional taxonomy. We all learned about taxonomy in elementary school and middle school. I have the breakdown down in the notes section here. Remember, we've all learned about kingdom and phylum and class and order, and down to genus and species. We categorize things with this system. But if you go to the very beginning of taxonomical organization, you find the domain. There are 3 domains that all living things can be assigned to. And again, within a domain, you're going to find multiple kingdoms. And within each kingdom, you're going to find multiple phylums, and so on and so on and so on, all the way down to the level of species, individual species. So if we think about those domains for a minute, we have bacteria, we have archaea, and we have eukarya. Within the eukarya, we find 4 kingdoms: animals, plants, fungi, and protists. Now, the way this picture is drawn here, it's drawn as a tree, if you will, a branching tree. This is called a phylogenetic tree. If you've had evolution, you know that we use a phylogenetic tree to show us evolutionary history in a diagram. We always start down at the base of the tree. That's where we imagine the very first life form, the very first cell. And across evolutionary time, it gave rise to the bacteria. That was the first domain to evolve on planet Earth, all of the bacteria. And then later on, the archaea evolved, and then the eukarya. So the eukarya, which is the domain we belong to, the eukarya is the youngest domain in evolutionary terms. Bacteria evolved first, so that's the oldest domain. And then sometime later, the archaea evolved, and then the eukarya. You and I, of course, belong in eukarya. So if we go back to that terminology of prokaryote, eukaryote, remember that's a way of describing cell types. The bacteria domain and the archaea domain are where all the prokaryotes are. Within the bacteria domain, all of the different types of bacteria, those are all prokaryotes. Single-celled, microscopic, no organelles, no true cytoskeleton. If you go to the archaea domain and all the many, many species in that, they are also all prokaryotes. They are prokaryotic, single-celled, microscopic, no organelles, no true cytoskeleton. All of the eukaryotic organisms are found in the eukarya domain. They can be single-celled or multi-celled, multicellular. The eukarya are much more diverse in terms of the types of structures of the organisms. So I'm not going to ask you to memorize the list of taxonomy categories. Don't worry about that. I do expect you to know that there are 3 domains of living things. And I expect you to know that the bacterial domain evolved first, followed by the archaea, followed by the eukarya. And I expect you to know that the bacterial domain and the archaeal domain are the prokaryotes, and that eukaryotes are all found in eukarya. Down at the bottom, kind of just for fun, again, I'm not going to test you on this. I broke down humans according to these taxonomical categories. A human being is found in the eukarya domain. We're in the kingdom Animalia. We're in the phylum Chordata. We're in the class Mammalia. We're in the order Primate. We're in the family Hominidae. I can never say this. Hominidae. We're in the genus called Homo and the species Sapiens. All living things can be broken down this way, that can be categorized and put into groups. Any questions about what I've said so far? Any questions about these terms, prokaryotic and eukaryotic, these cell types, or the domains that we find on our planet? A domain is going to contain many, many, many organisms that are all closely related to each other evolutionarily. So even though there are millions of different bacteria, they're all very closely related to each other from an evolutionary standpoint. And even though you might not think that you are closely related to plants and fungi and protists, you are more closely related to them than you are to, say, bacteria or archaea. Now on this slide, there's a diagram that I want you to put a star next to, because this is something you may not have given a lot of thought to before, and that is the relative size of different cells. This becomes more and more important as we start to use microscopes. We have to have an understanding of what we are looking at under a microscope based upon this comparison of cell size. So if you look at the axis down here, this ranges from as large as 1 centimeter all the way down through millimeter and micrometer and nanometer and down into angstroms. So we've got a huge range of size here. This cell right here on the right, this represents a plant cell. And of all of the cells, plants are the largest. Plant cells are the largest. Now, there are many different kinds of plant cells. So what we're looking at size-wise here is really an average. In other words, when we think about plant cells, we should be thinking about cells that are in the 100s of micrometers. Some plant cells might only be 100 micrometers. Some of them might be 200 micrometers, but they are generally within that range, the hundreds range. Now compare that to an animal cell. Animal cells are typically, again, on average, about 10 times smaller than plant cells are. Animal cells are more like tens of micrometers in size. So the two biggest cells are the plants and the animals. Now, if we go down to a bacterial cell, and you could also add archaeal cell here, prokaryotic cells. Now we're talking about cells that are in the single micrometer range. So a typical bacterium, it's hard to say typical when it comes to bacteria, but on average, a bacterial cell is going to range from about 1 micrometer in diameter to about 4 micrometers in diameter. So again, about 10 times smaller than animal cells. Now, why do we care about that kind of comparison? Well, when I'm looking under a light microscope at material that I've collected from the environment somewhere or from an organism, I need to know what it's possible for me to see. Plant cells and animal cells are relatively easy to see under a light microscope. In fact, if we go all the way up to the most powerful lens on a light microscope, we can see quite a bit of detail in plant and animal cells. It's different for bacteria and archaea. You can see bacteria and archaea under a light microscope, but you're not going to see them in any detail. You'll be able to see their relative size and you'll be able to see their shape. But that's about it. And that's an important thing to know. You would actually have to go to a much more powerful microscope, like an electron microscope, if you wanted to see any of the details of a bacterial cell or an archaeal cell. Plant and animal cells, they're great under a light microscope. We can see a lot under the light microscope, but not the bacteria and the archaea, the prokaryotes. You need a more powerful scope. Now, if I pull this picture back up again, look what's next on the size comparison. Here are viruses. Now, viruses are not cellular. Put a note or a star next to that in your notes, please. Viruses are not cells. They are particles. So by definition, viruses are not living organisms. They don't meet any of the definitions that we have for living things. They don't meet our cell theory. Remember, we said all living things are made out of cells, and this thing is not a cell. Now, this is a particular kind of virus drawn here. There are many, many different kinds of viruses, but it's important for us to note that viruses, viral particles again, are actually on a very, very small scale. Viruses range from about 10 nanometers to about 100 nanometers. Remember, for the cells, we were talking in the micrometer range. Viruses are 1,000 times smaller than that. All right, these are particles of material. These are pieces of genetic material, sometimes DNA, sometimes RNA, that are wrapped in protein. They are not cells, and therefore they are not considered to be living things. Very, very strange little thing, a virus. So I saw a hand go up. Sam, you want to ask your question?
Yeah, couldn't it be debated? Because like in other classes, we've talked about it. Couldn't it, even though they aren't made out of cells, but couldn't it be debated that they are living things?
Well, Sam,
They do
when
fit
I
into
say,
a lot of the categories for being a living thing.
so we're going to talk a little bit about the things that all cells have in common.
Okay.
It's a pretty short list, actually, and we'll talk about it in a couple minutes. There are certain characteristics that all cells have in common, prokaryotic cells and eukaryotic cells.
Yeah.
Viruses don't have any of those.
Okay.
Then there's a longer list of functions that cells have, and viruses don't have those either. Now, let me tell you why we sometimes think of viruses as being alive.
Because they can reproduce, right?
Actually, they can't reproduce. I know I'm blowing some of you away today, but viruses are what are called intracellular parasites. What happens in nature for a virus to be replicated, for a virus to be reproduced, it has to go into a living cell. And what's going to happen inside that cell is the cell is going to make copies of the virus. It's a parasite. It does not have the ability to copy itself because it doesn't have the things that are necessary to make a copy of itself. It doesn't have the enzymes that are necessary to copy its components. So what it does is it hijacks a living cell. It binds to that living cell It comes into the cytoplasm of the cell, and then the cell does the work of copying its genetic material and producing the protein that will cover it, that will make a capsule around it. It's fascinating and obviously very timely that we're in a pandemic. But no, viruses are not cellular. And by the cell theory, they are therefore not alive. Now, obviously, depending on which branch of science you're talking, depending on what kind of class you're in, it's not uncommon for people to talk about viruses as though they were alive. But in actuality, they're little particles. That's it. They're pieces of genetic material that are wrapped in protein. They don't think, they don't feel, they don't respond to stimuli, they don't reproduce themselves. They are simply inanimate particles. And the reason they're so problematic is because our cells don't know not to copy them. When they come into our cells see them and think, oh, here's a piece of genetic material. I should copy it. Here are some instructions for making protein. I should make some protein. And our cells end up doing the dirty work for the virus. So it's quite fascinating. Viruses are completely dependent upon living cells in order to get replicated or reproduced. We are gonna, we talk a little bit about viruses in this course. We don't talk a lot about viruses, but if you're interested and you'd like to learn more about viruses, we do have a couple of different microbiology courses here at Keene State that have a virology unit in them. What I'll do this semester, though, whenever we can, is I'll bring viruses in to our discussion so that we can compare them in terms of how they function compared to how cellular life functions. It's so interesting to me when I listen to certain people talk about the pandemic, when I listen to non-scientists talk about the pandemic and the SARS-CoV-2 virus, you often hear them refer to the virus as though it is a sentient being. For example, you'll hear people on the news say things like, this virus needs to adapt. This virus needs to mutate in order to survive. You'll hear language like that all the time. And in reality, none of that is true. Viruses exist because there are cells to copy them. And viruses mutate because they have genetic material, and all genetic material mutates over time. So yeah, it's very interesting, the language. The language is very interesting around viruses.
Professor?
Yes.
So I've had this debate in a couple different classes, are they alive? Are they not? What are they? That sort of thing. And one thing I never understood, because I've had some professors say they are live, and others say they aren't. So I can see why they're considered not to be alive, but how would they know what to do and how to find a cell and how to reproduce and how to lyse and do all that stuff if they're not alive?
So this is what's really creepy about viral particles. They don't purposefully do anything. So when this virus, this SARS-CoV-2 virus first emerged, and remember, we believe it came out of bats. Imagine that there was a mutation event, or actually several mutation events, and some bat somewhere breathed out the particles that we know as SARS-CoV-2. Now, we don't know a lot of the history around how this happened, but we do know that there was a human in Wuhan, China, who breathed in a group of these particles. Now, there is some evidence that perhaps it happened in a cave where a lot of bats lived. We don't really know, though. We don't know the true origin of this virus yet. That human being randomly took a breath one day and happened to catch viral particles in that breath. And that accident set off the chain of events that created this pandemic. Because that virus was brand new. That virus had not existed on the planet before. It was so mutated from its original form that it had never existed, essentially. That human with the viral particles inside their nose and their mouth was then the first basically incubator for that viral particle to get copied. The virus sticks to the cells only because it can. The protein on the outside of the viral particle has a shape. And in the back of your nose and throat, your cells have receptors that just fit that shape. And when that particle happened to accidentally, randomly collide with the cell stuck to it and took it in. And that started the process of making copies. And then that person sneezed and coughed out many, many copies. And the people around him or her started breathing them in. Because every person on Earth was vulnerable to infection with SARS-CoV-2, within a couple of months, it was an epidemic in Wuhan, China. Every person who breathed it in then made millions of copies of it and spewed them out, infecting more people, who infected more people, who infected more people. It then spread from China to Europe, to the United States, to Canada, to Africa, around the world, because every person was naive immunologically to that virus. It is an absolute random event when a pandemic occurs. You have to have an existing virus, an existing viral particle that mutates to the point where it is essentially a brand new particle. It's so different from the old particle that no one's immune system has ever seen it before. And so every person is susceptible to infection. Every person on the planet is an incubator to make millions and millions of copies of it. It is an exponential event. You start to see an epidemic in one area, then you get an epidemic in another area, then another, and then eventually it becomes global. It's fascinating and it's horrifying at the same time, but it absolutely begins with one person breathing in that mutated particle. That'll give you nightmares. That'll give you nightmares, won't it? So when you take courses or when you read source materials that talk about viruses as being alive, it's really more because we don't have language. We don't have a comfortable language to talk about that pathogen other than to think of it the way we think of other pathogens, the way we think of other things that can infect us and cause disease. But scientifically, on a pure technical scale, they're not alive because they don't meet the definition for living things. They don't have any of the structures that living things have on them. It's fascinating and it's bizarre but they are not cells. They are particles. They're particles of protein that have a little sliver of genetic material inside them. And of course, the SARS-CoV-2 virus that we're all battling has a piece of RNA inside of it instead of a piece of DNA. It doesn't have DNA in it. It has RNA in it. That's another bizarre thing. All of the cells that live on our planet have DNA as their genetic material. Some viruses have DNA in them, but other viruses have RNA in them, which makes them even more bizarre, even more strange. Good questions. All right. So the thing to remember from this slide about the viruses, again, we're talking about nanometers, between 10 and 100 nanometers in diameter for these viral particles. They are not visible under a light microscope. You cannot see a viral particle under a light microscope. So when you're looking at a slide under a microscope and you see a teeny tiny little dot, you might be tempted to think, oh, maybe that's a virus. It can't be a virus because viruses are just too small to be seen with that level of magnification. Now, if you go down below the chart, you'll see that there are these two pink bars. They're hard to see. The color is very faint. But this one is the bar that shows the range for a light microscope, and it ends right here. So anything within this size range, if we go up to the bar here, anything from here upwards, we can see with a light microscope. Anything from here down, you need an electron microscope to see it. So we can visualize viral particles, but we need an electron microscope to do it. And I know you've all seen that now very famous picture of SARS-CoV-2 with the spike proteins coming out of it. That image came from a light microscope. I mean, an electron microscope. Much, much more powerful instrument. Now, cells vary very widely in two ways. They vary in their size and they vary in their shape. What you're looking at on this slide is just a drawing of a handful of different cell types. And what we can take from this drawing is how differently shaped they are. Cell shape goes hand in hand with the function that that cell is trying to perform. Cells are shaped so that the job that they're doing is easier to perform. Now you see the shape and function relationship much more clearly in a multicellular organism, because each type of cell in a multicellular organism can have its own specialized function. So for example, muscle cells, which we call fibers, are quite elongated. There's 3 of them in this drawing right here. They have an elongated shape. And the reason that shape is important for a muscle is because it allows it to contract and relax, to get shorter and then get longer. It gives multicellular animals the opportunity to move their skeleton using these particular cells. This cell right here, this drawing represents a neuron, and neurons have these very extensive membrane extensions. Here's the body of the neuron. You can see it's got a nucleus in the middle, and then its membrane just goes outward into these little spiky things called dendrites. This is related to the function of a neuron. A neuron has to collect information from the environment and then send that information along to another part of the nervous system. So all these little spiky projections of membrane are reaching out into the environment to gather information. Over here on the right, this is an electron microscope image of two epithelial cells side by side that are found lining the intestine. And as you can see, here's one cell here on this side. And then here's the other cell over here on this side. As you can see, on this area of the membrane, there are also membrane extensions, but they're all very neatly organized into a row. These are called microvilli. They're extensions of the membrane. They look like little fingers. And what those microvilli do for these intestinal cells is absorb. This is a lot of surface area. All these folded finger-like projections give these cells enhanced surface area. And they need that surface area because their job is to absorb the food that passes by. So the shape here, this folding of part of the membrane, allows these cells to do that job. We said all cells have certain things in common. So it doesn't matter if you're talking about a prokaryotic cell or a eukaryotic cell, there are certain things that they have in common. There are structural things and there are functional things that all living cells are able to do. Now, if you look at this list here, this is more, this is sort of a combination of structural and functional things. We'll get to that. But first, I want you to look at this list. It's much shorter. These are the 4 structures that all cells have. All cells have what's called a cell membrane. They are all wrapped in a membrane. You'll hear it called the cell membrane. You'll hear it called the plasma membrane. The idea is the same. Those terms mean exactly the same thing. The cell is surrounded by a membrane. It defines the cell from everything that's outside of it. Inside of cells, you'll find this watery, sort of gelatinous material called cytoplasm. All cells have cytoplasm. All cells use DNA as their genetic material. And all cells contain structures called ribosomes. And ribosomes are just little machines, basically, inside of the cell that help produce protein. Remember, DNA is a piece of nucleic acid that contains a code. And that code is basically to build protein. The ribosomes do the work. They do the work of building the protein. All cells have these 4 universal structures. Now, if we go up to this list, you'll see a few more things. Again, surrounded by a plasma membrane. Yep, we already said that. Store genetic information in their DNA. Yep, we said that. But here are some functional things. They're able to use a process called semi-conservative replication in order to copy their DNA. Remember, cells divide. That's how you get new cells. And every time a cell divides, it has to copy its DNA molecule so that the 2 new cells that are going to be produced each get their own copy. That process of copying the DNA is known as replication. All cells are capable of replicating or copying their DNA molecule. All cells are also able to do something very different with their DNA molecule. They're able to use it to make protein. Living cells are able to do a process called transcription in order to take the DNA molecule and make an RNA molecule from it. They're also able to do a process called translation, where they take that RNA molecule and they use it to build a protein. That's where those ribosomes come in. Living cells have additional proteins in them that they use as enzymes. And finally, living cells contain 4 what we call macromolecules. These are protein, carbohydrate, lipid, and nucleic acid. All cells have those 4 sort of building block macromolecules inside of them. Now, one of the questions that has been asked for a long, long time is, where did that very first cell come from? How did that very first cell emerge on planet Earth? We know that in the very earliest time of this planet, the very earliest history, the Earth was a very inhospitable place. It was very hot. It was volcanic. There was toxic gases everywhere. It wasn't exactly a place for life to suddenly emerge from. It was also completely water covered in those days. The land masses had not emerged yet. So for a long time, scientists have asked themselves, how could a cell have come about? How could a cell have first emerged through evolution on this planet? There was a very famous set of experiments that were done by scientists who were called Miller and Urey. This was back in the 1950s. And what Miller and Urey were able to do was essentially recreate in the laboratory the kinds of conditions that would have been found on that early planet Earth. And what they demonstrated in this experiment was that if you combined all those things, all the things that planet Earth looked like in those days, you could actually show the production of small molecules that could have then combined to create the first cell. They called this process chemical evolution. So remember, early Earth was a very different place than it is now. It was covered in water. It was full of lots and lots of different gases, including toxic gases. So hydrogen gas and methane and ammonia and so on. The sun was certainly available. The sun was producing light energy onto the Earth. There was heat available, and there was electricity available because of the storms that would have been occurring in the early atmosphere. So what Miller and Urey did was they set up this sort of an artificial Earth in the laboratory using just glass apparatus. They combined water with gases and sparks of electricity in order to see what would happen. And what they discovered was that these elements, these very simple elements like carbon and nitrogen and hydrogen and oxygen would have come together to create small molecules that were a little more complex. Sorry, I didn't mean to advance that slide. A little more complex than these very simple gases. And what Miller and Urey suggested was that as these very small elements combined to create small molecules, maybe those small molecules then combined with each other to create the macromolecules that later became cells. Now, there's been a much more recent advancement in our understanding of where cells came from, and that was back in 2009. There was an experiment done by a scientist or a group of scientists led by a scientist named Sutherland. And what Sutherland did with his group was basically expand on what Miller and Urey had done. They recreated that sort of all those conditions that the early Earth would have had. And they discovered that if you introduced some very small carbon-containing molecules into that system, you could create at least a partial ribonucleotide. Now remember, a ribonucleotide is the building block of RNA. Sutherland's group showed that by setting up the Miller and Urey experiment, they could create a piece of that base called cysteine. They could create a piece, a part of the cysteine base inside that apparatus. The other thing they found was that that partial cysteine molecule could combine with other chemicals, other gases, to create the, I keep calling it cysteine, I apologize, cytosine, to create the whole base called cytosine. Remember, in DNA and RNA, there are bases, there are nucleotide bases. DNA has adenine, thymine, guanine, and cytosine. RNA has adenine, uracil, guanine, and cytosine. What Sutherland's group proved in the lab or demonstrated in the lab was that setting up the early Earth, setting up that chemical evolution experiment, they could create cytosine. They could show that cytosine would emerge in the test tube. They then took that test tube apparatus and exposed it to ultraviolet light. And that cytosine produced guanine. Sutherland's group showed how 2 of the 4 bases inside nucleic acid could have evolved on our planet. They took the next great step in showing experimentally where life came from on our planet. And it originally came from all of the other pieces and parts that were present on Earth all those billions of years ago. Heat, light, particularly ultraviolet light, electricity, and gases, and water. When you put all of those things together, you can create what is really the essence of living things, which is DNA and RNA. If you're interested in learning more about Sutherland's experiment, the program called NOVA, this is a program on PBS. They did a special called, the program is called NOVA Science Now. And they did an episode called Where Did We Come From? And this is a link, you'll have to copy it and paste it into your browser. But this is a link to that program that NOVA produced about these groundbreaking experiments. So Miller and Urey and Sutherland certainly showed how life could have started on an early planet Earth, but no one has shown yet how the first cell came about. Remember, a cell has a certain set of structures, right? It has to have a membrane around it. It has to have cytoplasm. It has to have DNA in it, and it has to have ribosomes. So we've not yet shown experimentally where the first cell came from, but other research has shown where something sort of like a cell could have come on the early Earth. In fact, this work is now considered evidence of the first thing on planet Earth that was capable of copying itself, that was capable of making a copy of itself. This thing is now known as a ribozyme. Remember, Sutherland's work showed how nucleic acid could have emerged, cytosine and guanine, two of the bases that we find in DNA and RNA. A ribozyme is essentially a piece of RNA that has enzymatic activity. A piece of RNA that is able to copy itself. Remember, that's a hallmark of a cell. The fact that it can copy itself, it can divide, and it can make new cells. So ribozymes are not cells, but we have evidence that a ribozyme could have formed on planet Earth. And from that ribozyme may have come the first cell. So work goes on in these laboratories to try to demonstrate how that first cell could have emerged. What happened that allowed that RNA, that ribozyme, to get wrapped in a membrane and develop cytoplasm and develop ribosomes? We don't yet know all the answers, but there is some exciting research going on all the time. Now, there's a couple of other things that I want to just introduce today as we talk about cells in our last 10 minutes or so. One thing we know is that all cells require energy. They require a source of energy in order to do the work that they do every day, to stay alive, to survive. And we know that if you look at any cell, there is a molecule that provides that energy in a stored form. And that molecule is called ATP. ATP stands for something. It's adenosine triphosphate. This is a universal storage form of energy. All cells use ATP to get energy when they need to do work. Now, where does ATP come from? Well, it comes from a couple of different chemical reactions. It comes from a type of reaction called cellular respiration, followed by oxidative phosphorylation. And it can also be produced by a process called fermentation. Different types of organisms produce ATP a little differently, but all cells use ATP for their energy needs. You should also note that there's sort of a cousin molecule to ATP, one that we're going to talk about a couple of times this semester. And that's a molecule called GTP. GTP stands for guanosine triphosphate. GTP is believed to be sort of the precursor to ATP. Earlier in our evolutionary history, cells probably used GTP. And then over time, ATP replaced GTP. You still see a little GTP used though, in certain cells, in certain reactions.
Professor?
Now, yep, go ahead.
During the oxidative reduction and whatnot, I know that that's one of the reasons why like you have to breathe, because it requires the oxygen to make ATP.
That's right.
And in another one of our classes, our professor was telling us about like the non-oxidative versions, which are like, they actually like, I guess they use your muscles to break it down. And she said it's pretty much like your extra energy boost to get away from the saber-toothed tiger that's trying to eat you alive. And
that's a good
where it came
analogy.
from, it was just to give you a little extra
That's
run
right, a
for
little
your
boost.
life.
That's right. That last little push that you might need.
Even when you couldn't, if you weren't taking in enough oxygen, there was one extra way to give your muscles just a little bit more energy.
That's right. You know, in humans particularly, and other animals, I'll say, we have kind of redundant systems, which is good because it helps us survive in sometimes very challenging circumstances. We make our ATP preferentially using that process called cellular respiration and oxidative phosphorylation. And yes, we do it aerobically. We need oxygen in order for that whole long series of reactions to occur. What's interesting is that we have some redundant systems though, when we can't do our preferred metabolic reaction. There are other things that we can do to get through a short period of difficult circumstances. For example, when a human is exercising beyond their capacity, when they're exercising to a level where they simply cannot breathe in enough oxygen to do cellular respiration normally, humans can actually switch and start doing a process called fermentation. We happen to do a type of fermentation called lactic acid fermentation. And if you have ever overdone it aerobically, like if you've ever gone for a run and you pushed yourself past your ability, you know, you probably gasped for breath while you were running, and you probably did not take in enough oxygen, and you probably fermented a little bit and produced a little lactic acid in your muscles. And lactic acid hurts. Lactic acid stings because it's an acid. So while you're running, you know, beyond your fitness level and you're breathing like that, you're also making lactic acid through fermentation. It helps us get enough ATP when we can't get enough oxygen to make ATP the normal way. The other process that you're referring to actually takes place in muscle cells, and it's a way for the muscle cell to sort of beat a system. One of the things that we're going to talk about a lot this semester is that cells are very thrifty. Cells do not store ATP. It seems bizarre. You would think that a molecule that is as important as ATP is, a cell would store, a cell would sort of hoard ATP for when it needs it. But cells are very thrifty, so they make ATP when they need it. And sometimes what happens, particularly in skeletal muscle, is that the demands for ATP far outweigh the rate at which new ATP can be made. And skeletal muscle has evolved to have a little backup system, and it involves a molecule called creatine. And what creatine will do is take that ATP molecule and steal one of its phosphate groups and creates a molecule called creatine phosphate. And it leaves behind the rest of the ATP molecule. It's called ADP. Now, when the cell takes stock, if you will, it's going to see that, oh, I used to have a little ATP around, but it's gone. I have ADP here. I don't have ATP here. And it'll start making more ATP. So what's happening in skeletal muscle is the creatine is sort of fooling the cell into believing that it doesn't have enough ATP around. And when the muscle needs to perform at its maximum, the creatine phosphate will give up the phosphate group, give it back to the ADP molecule, and suddenly have more ATP. So human beings have evolved redundant systems for ensuring that we have enough ATP under even some very, very challenging circumstances, even when we can't get enough oxygen to meet our needs. Great question. All right, we only have a couple minutes left, so this is a good place for us to stop today. Remember, we're going to get together on Thursday. We'll finish up what we were talking about today, and then we'll talk about the science of biology. We'll move forward into our next lecture topic as well. Once we finish Thursday's lecture, you'll be ready to try the first lecture homework. You don't want to try to do that lecture homework though until we've had our lecture on Thursday, because you won't have enough information yet to complete it. Okay, so between now and Thursday, if any other questions arise, any questions about the syllabus or the schedule or anything we talked about in lecture today, feel free to message me. Message me through Canvas, and we'll work through those questions. And otherwise, plan to see me on Thursday over Zoom. Okay, everybody? Thanks for coming today. Enjoy the rest of your day or your evening, I'll say. And I'll see you again on Thursday.
Thank you.
You're
Thank
welcome.
you.
We'll see you
Have
next
a good
time.
day.
Thank you.